Hello students, this is a recorded lesson um, for on Tuesday. Okay, I'm actually recording it tonight, which is still on Monday. Okay, so we have covered learning objectives number 11, and now today's learning objective is the next one, which is number 12. Okay, explain the sliding filament model. Okay, sliding filament model of muscle contraction, including the roles of troponin, tropomyosin, calcium, as well as ATP. So there are four things that you need to know, yeah? The role of troponin, the role of tropomyosin, the role of calcium, and the role of ATP. Okay, so today's lesson is only about this learning objective number 12, which is actually quite a lot already. I have updated the slides. Okay, so I will share to you the latest one through the WhatsApp group. Okay, the PDF form of this lecture. Okay, so yesterday we talked about the sarcomere, which is the structural unit of myofibril. I hope that you know already the different features, yeah, of a sarcomere, and of course the labeling, the Z line, the M line, the I band, the A band, and the H zone. Yeah. Okay, and of course the myosin, and then the actin filament. Okay, so let's just have a look at this particular image. Okay, this is okay. First of all, what are we looking at here? First of all, we are looking at myosin. Okay, as you can see, this is thicker. Okay, it's quite thick. And then this is actin. It's a bit thinner. Okay, and of course the myosin has what we call this myosin head. Yeah, myosin head. So this is called myosin head. Okay, and then we looked at the tropomyosin. So this, you can see this tropomyosin here. So this is tropomyosin. This is a blocking protein. Okay, but let's just look at, it's quite a long protein. Yeah, it's pretty long protein. Okay, as you can see there. So this is a tropomyosin. Okay, and then troponin. So this is troponin. This part here is troponin. Okay. So remember, this is the troponin. The troponin is where calcium ions will bind. Okay. And then this is the tropomyosin. Tropomyosin is also a type of protein that actually blocks this myosin head from binding to its binding site. Okay, so this is an actin filament. There are binding sites for this myosin head. Okay, so the actin filament contains binding sites, okay, for this myosin head, which is actually at the moment is blocked here. It's blocked by this tropomyosin. So for example, like this one here, the binding site is just under, is covered by this. It's actually under, it's actually covered by this tropomyosin. Okay, that is where the binding sites for the myosin head is, which is currently blocked. Okay. Okay, so we looked at the actin, we looked at the myosin. So when we look at those structures, we have to also look at this structure of this sarcomere. Okay, like this. Okay, so the myosin head is actually not shown here. Yeah, what all you see here is just myosin and then the actin. Yeah, the myosin head is actually not shown here, and you will be able to see the myosin head if we zoom out like this one here, as you can see now. Okay, so now you can see the myosin head here. Okay, so these are the myosin head, this is also the myosin head. Yeah. Okay, right, so we have to learn the features first. Like I said, you need to know troponin, which is the protein where calcium binds to. Yeah, calcium will bind to this troponin, and then tropomyosin is a blocking protein. What does it block? It blocks the binding sites for this myosin head. Okay, so I repeat, this is called myosin head. Okay, the head of the myosin. So at the moment it is in a resting state. Yeah, resting state. And so when action potential occurs, okay, remember when action potential occurs, when the action potential travels down the T tubules, yeah, the sarcomere will release the sarcoplasmic reticulum. The sarcoplasmic reticulum will release calcium, yeah, into the cytoplasm or sarcoplasm. The cytoplasm of muscle is called sarcoplasm, and so the calcium will bind to troponin. As you can see here, the calcium will bind to troponin, and when this happens is that conformational change occurs. Conformational change occurs. Now this is something which is we are already familiar with this term. Yeah, conformational change occurs. Okay, we have seen this term conformational change in cell signaling for glucagon hormone, and now we come across this term again, conformational change occurs. Basically, what does it mean? It means the shape changes. Okay, in this case, the tropomyosin. Yeah, so now the tropomyosin will move. So this tropomyosin will move. Yeah, will change its shape. In this case, it moves away and to expose the binding sites. Okay, so the binding sites now is this one here. This is the binding sites for the myosin head. Okay, so when the tropomyosin moves, it will reveal. Yeah, it will reveal the binding site. Is also another binding site here. It will reveal the binding sites of this myosin head. Okay, and now the myosin head can now form cross bridge. Yeah, and we call it here actomyosin cross bridge, and there's also another actomyosin cross bridge. Why it's called actomyosin cross bridge? Because it is like a bridge, yeah, that connects between myosin and actin. So it's called cross bridge because it's like crossing. Yeah, crossing. Okay, so so if you read here, calcium ions bind to troponin, the troponin changes shape. This causes troponin and tropomyosin to move away from the myosin binding site. Myosin heads can now bind and the muscle fiber can contract. So the way this muscle fiber contracts is that the myosin head will pull this. So it's actually pulling, yeah? Pulling this in this direction. Okay, it pulls in this direction or tilts. Yeah, tilts T-I-L tilts, T-I-L-T-S. Okay. In this, for example, in this case, it's pulling in this direction, in this direction, in this direction, and in this direction. Okay, so that's the pulling. Okay, now, so this is just an overview, yeah. So what we have seen here is an overview, just to trying to introduce to you the names like tropomyosin, the troponin, and how does the myosin head form the actomyosin cross bridge, and how does it contract? It contracts by actually pulling the actin filament. So this particular actin will glide past each other, yeah. For example, this one will glide in that direction. Will also glide in this direction. Okay, so that's why it's called the sliding filament theory, yeah, the sliding filament model. Okay, now let's have a look at this one here. This is from another textbook. Okay, now that you can see the M line, yeah, the M line, and this is one. The Z line is not shown here, but you can see the M line right in the middle here. Okay, so when the muscle is relaxed, tropomyosin and troponin are sitting in the position in the actin filament that prevent myosin from binding. So prevent myosin head, yeah, to be specific, prevents myosin head from binding. Okay, so at the moment, as you can see here, it is detached. This is detached. Okay, detached. It's not connected yet. Okay, and the next one here, when muscle contraction starts, the troponin and tropomyosin change shape to allow myosin to bind with the actin. So this change shape here is what we call just now conformational change. Okay, conformational change. Okay, now if today we're not going to show you the video because the video is meant to be on Wednesday. Yeah, I will show you the video, the link to the video of this sliding filament model. But you know, just if let's say if I were to label this 1, 2, 3, 4, 5, 6. Okay, and let's say 7, and then 8, and then 9. Okay, in terms of region of the actin. So if this you know, like rowing, when you row a boat, you're rowing, because you will not understand until you watch the video. But before you watch the video, I'm just going to show you how this actually works. Yeah, so when this thing here pulls or tilts, yeah, in this direction. Okay, so basically this whole thing is shifted. Yeah, this actin filament is shifted. So if it pulls, so that means now the number 7, so now this one becomes number 2, 2, 3, 4, 5, 6, 7. Okay, and then 8, and then 9. So when it pulls again, contraction occurs here. So later on, the number 8 will be, the next one will be number 8. So it's very difficult to actually show you here, but if you can understand what I mean. So next one, this one will become 3, 4, 5, 6, 7, and 8. Yeah, basically this actin filament will move closer and closer. Okay, one at a time. So the video will show you what I mean by this muscle contraction. Okay, the rowing motion. Okay, rowing motion. Okay, I think that's about it for that particular slide. And so as you can see here, myosin held tilts, so the word tilts, okay, pulling the actin and causing the muscle to contract by about 10 millimeter. And then it will move again 10 nanometer, sorry, 10 nanometer, and then it will move again by 10 nanometer and 10, another 10 nanometer and so on. Yeah. Okay, so basically this will, the actin filament will move. It's actually moving the actin filament one at a time. So this segment will be pulled, okay, and then another new segment will be pulled. Okay, meaning when it's moved here, then it will pull the next segment. Okay, and then it will pull the next segment, if you know what I mean. Okay, until now, how long does this contraction occurs? For as long as the ATP is available and for as long as calcium ions are available. Sorry. Okay, ATP hydrolysis causes the release of myosin head. They spring back and repeat the binding and the tilting process. Now this ATP, we're going to talk about this ATP in a moment. It's very, very important. Yeah? It's very, very important that you know the role of this ATP. Now, as you can see, I have referred to three different textbooks, yeah, just to explain this sliding filament model. Now, this is the most important one, the last reference coming from another textbook, okay? Okay. Now, because it is a model, you can actually, this model can start at any point, yeah? Meaning you can begin when the head is relaxed. For example, in the previous, here, in this section here, it begins where the myosin head is still detached, okay? Still in a relaxed mode. In this particular model, it's actually also in a relaxed mode. The head is still detached. In this particular textbook, it starts with the head is already bound, yeah, is bound to the head is actually has bound to the binding sites. But of course, you have to know this particular textbook, this particular slide, it will show you the importance of ATP. Okay, the importance of ATP. Sorry. But of course, before the head binds to this binding site, you have to remember the role of calcium. Okay, remember the role of calcium, which binds to troponin, and then the role of tropomyosin. Okay, so this is not mentioned anymore. The calcium, the troponin, and the tropomyosin is not mentioned here because it's already mentioned in the previous slides. So now the focus is the ATP, the ADP, and the phosphate group. Okay, so yeah, before this, of course, the calcium binds to troponin, the tropomyosin will move away and it will expose the binding sites of the for the myosin head. So now the myosin head is already bound to the actin. Okay. Now, when it's already bound like this, the ADP and phosphate group is attached. This is attached. So in this particular state, it's already attached, okay? Now, the release, when the ADP and phosphates are released, okay? When this ADP and phosphate are released, this is actually the power stroke. Yeah, the power stroke means the tilting process, the pulling process. Okay, so when ADP and the phosphate groups are released from the myosin head, this is actually the power stroke, meaning now it can pull, it can do the pulling action or tilting action. Okay, as you can see here, myosin head tilts, which forces the actin to move in relation to myosin. ADP and phosphate are released. Okay, and this is the power stroke. The power stroke. So the power stroke is obtained when ADP and phosphate group are released from the myosin head. Okay, from the myosin head. So once this is already the ending of the pulling process, okay, it still remain bound, yeah, remain bound, the head still remains bound to the binding site. Now, because the ADP and phosphate are released, now ATP can now bind. ATP can now bind, okay, and then the ATP will be hydrolyzed. So once the ATP binds to the, when the ATP binds to the myosin head, this ATP can now be hydrolyzed back to ADP and phosphate group by enzymes ATPase. Okay, now this enzyme ATPase, this enzyme ATPase is actually found on the myosin head. Okay, in fact, the textbook says the myosin head itself is the ATPase. Yeah, or simply we can put it in a simple way that the myosin head, has the ATPase enzyme. Okay, so once the ATP binds to the myosin head and then it's hydrolyzed, then that will provide the energy. So let me just repeat here again: ATP, ADP plus phosphate group plus energy. Okay, this is ATPase. Now this will provide the energy to detach, the energy to detach this head and then to return back to its original position. Okay, to return myosin head to previous position. Okay, so a lot of students think that this pulling effect is the one that requires energy. Yeah, a lot of students think that when it pulls or when it tilts, okay, this requires the hydrolysis of ATP. Now that is not the case. Okay, that is not the case. Okay, in fact, the detachment of the myosin head from its binding site and then the return of the myosin head to its previous position, these are the two reasons why energy is required. Okay, energy is required in this process. So if we have a look here, the myosin head can only be released from its binding site when ATP is hydrolyzed to ADP and a phosphate group. The myosin head contains ATPase enzyme. In fact, your textbook mentions that the myosin head, the myosin head is an ATPase. Okay, but I think to put it in a simple word, let's just say that the myosin head contains the ATPase enzyme. So hydrolysis of ATP by ATPase releases energy. So remember, releases, yeah, release. Remember, when when bonds form, energy is stored in the bond, and when the bond is broken, energy is released. Okay, so we use the term release. Hydrolysis of ATP by ATPase releases energy to detach. So this is the number one, for detach the myosin head, and then to reposition. This is number two. Okay, to reposition. This is number one, and this is number two. Let's just use a clearer. Okay, detach. To detach the myosin head, that's number one, and then to reposition the myosin head to its previous position, and this is where energy is required. Okay, right, and so you can see now that it will go back to its, this is already the previous, this is already the original position. This is already the original position. Okay, the myosin head hydrolyzes the ATP to ADP and phosphate group. This provides the energy to release the myosin head from the actin. The head flips away. Flips away means return back to its original position. Okay, so you can actually start, if we can rewind a little bit. Yeah, it is better, of course, when you watch the video, but there are many videos out there available, yeah, on YouTube, but you have to be very, very careful, yeah, because sometimes those videos are made by students, so the information may not be valid. So it can be wrong. So if we look at this particular slide again, we starts from number one, number two, and then number three, and number four. Okay, now you can actually begin this as the first one if you want. Like, for example, this is the first one. Okay, this is the first, where it is still detached. Okay, the head is still detached, the myosin head is still detached, and the ADP and phosphate group are still detached. And then you can go, now it is bound. This is number two if you want. Yeah, this also can be done. Now this is number two. Now that it is bound, but of course, in between, you need to talk about the role of calcium ions, the role of troponin, and the role of myosin. Yeah. Okay, so now it is detached. Calcium binds with troponin. Tropomyosin will move away, and so will expose the binding site. Okay, and so the myosin head can bind with the binding site. Okay, now and then now this will be released. The ADP and phosphate will be released. Okay, and that will give that power stroke. Okay, power stroke meaning the pulling effect, pulling or tilting. Okay, pulling or tilting. Okay, now you can see here it's released. So when it is released, now ATP will bind. ATP will bind, okay? And so, in order to detach this, myosin head, the ATP must be hydrolyzed, okay? So again, so once it has pulled, once the pulling process is done, the tilting process is done, now this must detach, okay? In order to detach this and to return back to its original position, this ATP must be hydrolyzed to ADP, phosphate group plus NAG, and then the enzyme is ATPase. Yeah, the head itself here, according to the textbook, is ATPase itself. Okay, is the enzyme itself. The myosin head is actually the enzyme ATPase. So the energy from the hydrolysis of the ATP release is used to detach this myosin head and will return back to its original position. Okay, and then the cycle will continue. Okay, so the important thing to note down here is the role of the ADP and phosphate group, and the role of ATP, and the role of the ATPase. Okay, so if we look at this notes here, the myosin head are now able to attach to the binding sites, forming ectomyosin cross bridge, pulling the actin over the myosin. ATP is needed. So this is now, just want to highlight this. ATP is needed to break the link and to allow the cycle to be repeated. ATP is required, meaning energy is required. The hydrolysis of ATP, what it means here is hydrolysis. Hydrolysis of ATP to release energy is required to detach the myosin head. Break the link means detach the myosin head, and to return the myosin head to its original or previous position. Okay, as you can see here, hydrolysis of ATP releases energy, which is used to detach the myosin and to reposition. Okay, and what happens during relaxation? During relaxation, there when there's no more stimulus, when nervous stimulation of the muscle stop, there's no more stimulus. Okay, that means there will be no there will be no action potential, there will be no release of calcium. The calcium will be pumped back into the sarcoplasmic reticulum. Okay, we pump back, which also requires the hydrolysis of ATP. Okay, so when with no calcium present, tropomyosin changes position and blocks the binding site for the myosin head, to the actin filament. The muscle is now relaxed and can be extended, meaning the relaxation can be further extended if the other opposite muscle contracts, because muscle works in opposite ways. For example, if biceps contracts, then triceps will relax, and vice versa. Okay, so the muscle is now relaxed and can be extended by the contraction of the opposite muscle, of the antagonistic muscle. Okay, I hope you understand that. I think this is from O level. All right, some notes here on tropomyosin. It says here a blocking protein. We know this already. When the muscle is relaxed, it blocks the binding site for actin filament. For the muscle to contract, tropomyosin must be moved. Yeah, remember the moved means conformational change. Conformational change. Calcium ion will help to unblock the binding sites by moving the tropomyosin. Okay, but of course before that the calcium ions will bind to, remember, binds to, it's not really clear here, this calcium ions binds to troponin. Okay, remember it binds to troponin first, only then the tropomyosin can be moved, which will expose the binding sites for the myosin head. Okay, and then the ATP. Now it's important again. When hydrolyzed, so it says when hydrolyzed provides energy. So actually, we don't actually like to use the word provide, but instead releases. Okay, releases energy to detach the myosin head from the active filament. So that's one role. That's what the energy is used for. And then to reposition of the myosin head, and as well as, which is not mentioned previously, as well as to for the active transport of calcium ions back to the sarcoplasmic reticulum. Okay, this is the third one. Okay, now to understand further, let's look at this dispositive paper here. Look at the number of marks here. 5 marks. It's staggering, 5 marks. Explain the role of ATP in the contraction of striated muscle. So we know about ATP. The role of ATP, as you can see here, so in order, when you look at the answer here, you have to explain the because you cannot just simply write down that the hydrolysis of ATP will release, will detach the myosin head from its binding site and will reposition the myosin head, to its previous position. You need to actually describe what happens initially. So as you can see here, myosin head binds to the actin, forming the cross bridge. ADP is released. Remember, when this is released, this is the power stroke. This is when it's able to pull or tilt. Okay, so first, of course, binding, the myosin head binds to the actin. Okay, done. And then ADP and phosphate group are released. That will help to tilt. Okay, remember this one here. This is release, and that will pull or tilt. This is the power stroke. Okay. And then the next one, ATP binds the myosin head. Okay. ATP detaches from the actin, but this is like it won't go. ATP binds to the myosin head. Before this detach, you have to mention ATP is hydrolyzed. Okay, which is this one here. Point number 7. Myosin head ATPase causes the hydrolysis of ATP. Okay. So ATP is hydrolyzed into ADP and phosphate group plus energy. So that energy is used to detach okay, detach, and then for tilting back to its original position. What I don't like about this particular answer scheme is that particularly this 5, 6, and 7 here is not actually written in a sequential manner. Yes, supposedly this is how it should look like. This is correct. Correct. Power stroke. Bind to the myosin head. Now before it detaches, this must be the next one. This must be number 6. This must be number 7. Okay. So when the ATP binds to the myosin head, the ATP is hydrolyzed. The ATP is hydrolyzed into ADP and phosphate group. The energy is used to detach the myosin head. Yeah, this is the first one. The energy is used for this, and the energy is used for this. Okay. So once the contraction is done, the calcium will be pumped back into the sarcoplasmic reticulum, which also requires the hydrolysis of ATP. Okay, so it's 5 marks. That's quite a lot of marks to score. Next one, describe what happens in the sarcomere. So sarcomere, when myosin head releases. So see, releases ADP. This is the pulling effect, pulling and the tilting of myosin head. In other words, this is actually the contraction. So what it means is that what happens to the sarcomere when it contracts. Okay, so of course the answer is shortens, and then the Z lines move close together. See, rowing motion. Rowing motion means the pulling, tilting of the myosin head. Okay. Power stroke. Yeah, that's the meaning of power stroke. Power stroke means the pulling effect, the tilting effect. Okay, and then precise. Explain the precise. Yeah. Looking for my pen, it's actually hiding there. So precise, yeah, precise function of ATP in the sliding filament mode. Let's look at the answers here because we learn by learning the answer scheme. Yeah, so as you can see here, the precise function of ATP. See, the ATP binds to the myosin head, and then the hydrolysis of this ATP by the ATPase. Remember, the myosin head itself is the ATPase. Will help to detach. Remember, the first function, detach, and then return back to its original position. So ATP binds the myosin head, hydrolyzed by ATPase, which is by the myosin head itself, into ADP and phosphate group and energy. That energy is used to detach the myosin head from the actin or from its binding site, and then to return back to reposition the myosin head to its original or previous position. Okay, so this is very, very serious. The ATP is somehow very complicated actually. Yeah, so you need to understand this particular slide here is very, very important for you to go through. Yeah, you can, like I said, if you may begin, if you don't like this is already bound, you may start with this one here, which is still unbound. Okay, this you can start this as your number 1 process, which I think I prefer that it is still not bound, the myosin head is still not bound, okay? But the ADP and phosphate are already attached, okay? And then when it's bound, because of the calcium, yeah, binds to troponin, tropomyosin move away, it exposes this binding site, so it will bind. So when it's bind, so this is number 2, okay? And then when this release, this will have the power stroke, the pulling can start, the tilting can start. That's when the ADP and phosphate are released, yeah? And so it's still stuck, yeah, it's still bound here. So to detach this, number 3, yeah, number 2, sorry, this is number 3, and this must be number 4, okay? So to detach this, the ATP must be hydrolyzed, okay, into ADP and phosphate group, and then the energy is used to detach, and of course to reposition this back to its previous position, like that, okay? So okay, so that's the role of a lot of students. Like, let me remind again, a lot of students think that the hydrolysis of ATP releases energy for the power stroke, yeah, and that is wrong. That is the biggest mistake for most students. So hydrolysis of ATP that releases the energy is not for the power stroke, yeah, it's not for the pulling. It's not for the pulling or tilting process, but rather the ADP and phosphate are released from the myosin head. Only then it can create the power stroke, okay, the pulling effect or the tilting effect. Okay, so go through again the past paper, which is very important. So now let's go back to here, okay? So now we can talk about now you should understand the next the events in the muscle fiber. Remember yesterday I mentioned about events at the neuromuscular junction. This is the same as in synaptic transmission, okay? But when action potential travels down the T tubules, okay, that causes calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin. Now you know what I mean by this step number 9. Calcium binds to troponin. Tropomyosin moves to expose the binding site. Myosin head forms cross bridge within filaments and the sarcomere shortens. So that's the meaning of that step number 9. Okay, now we are almost towards the end here. So ATP, where does it come from? Of course, it comes from the cytoplasm or sarcoplasm. However, for muscle contraction, muscle also have stored or source of ATP, and that is from creatine phosphate. So creatine phosphate provides alternative source. Alternative source means not the main one, but in case if ATP is run out, okay, the ATP is running out, so it will resort to creatine phosphate as a source of ATP, okay. It's also found in sarcoplasm. Sarcoplasm means cytoplasm of muscle fiber. It is the immediate source of energy once they have used the ATP in the sarcoplasm, or used up, yeah, or finished. Okay, so this is how the process is done. Creatine phosphate plus ADP to give creatinine and ATP. Yeah, because the phosphate can be removed. This phosphate can be removed and then react with the ADP here, okay, to form ATP. This is removed. The creatine, the phosphate from creatine is removed. The phosphate binds with the ADP to form ATP. Okay. And this is the reverse. So this is the reverse. So this process here is reversible. This is reversible. Okay, reversible. Okay, I think that's about it. Yeah. So I think remember we just stick to the according to the learning objectives here, the roles of troponin, which is for calcium to bind. Calcium will bind to troponin. That will trigger that will remove or that will cause conformational change, meaning the tropomyosin will move away, exposing the binding sites for myosin head. The calcium is for binding with troponin. The ATP has a lot of roles in this case. Yeah, the first one was for detach. The second one was for repositioning. The third one was the pumping of calcium. Yeah, pumping back the calcium into the sarcoplasmic reticulum. So let us look at that one more time here. Okay, remember when action potential occurs, the calcium is released. Calcium is released. Yeah, this is of course by facilitated diffusion, and then the calcium will go and find troponin, binds with troponin. Yeah, and then that will cause the tropomyosin to move away, and so on and so on. Okay, so once contraction has finished, okay, this calcium must be pumped back. Yeah, it is pumped back into the sarcoplasmic reticulum because it's against its concentration gradient. So that's where the ATP is required as well. Yeah, ATP, hydrolysis of ATP is required to release energy for the pumping process. Okay, so hopefully this you managed to understand. Yeah, the textbook, your main textbook uses this one here, which is not really to me it's not really complete. Yeah, especially it doesn't show the role of ATP and ADP. Yeah, and phosphate group. So in your main textbook, this is what is given to you: this one, as well as this one, which is for me is still not enough. But this particular textbook is very good because they show you how the release of this. It shows you the release of the ADP and phosphate group. Okay, will cause the pulling process, the power stroke, the tilting process, and then the binding of the ATP. So once the ADP and phosphate group are released, the ATP can now bind. Yeah, can now bind. And this is getting ready for the detachment. Detach or remove to release the myosin head requires the hydrolysis of ATP, and to reposition this myosin head back to its original position. That also requires ATP. Okay, so this is very, very important. The concept is very, very important. The hydrolysis of ATP is not for the power stroke like many students think. The hydrolysis of ATP is not for the pulling process, it's not for the tilting process, it's not for the power stroke, but rather the ADP and phosphate group are released, then this causes the pulling of the actin filament by the movement of this myosin head. Okay, so that's very, very important. Yeah, I hope I have answered. I have managed to answer this particular learning objective. It's not easy to answer this particular learning objective. Yeah, 12 year, because of the notes which are given to you. Yeah, from the textbook is not enough. Yeah, it's actually not enough. All right. So on Wednesday alone, this is for Tuesday's lesson. On Wednesday, I will give you the link here to watch more on the sliding filament model. Um, but you have to wait for the link that I will give to you on Wednesday. Okay, because there are many videos out there on YouTube which are not correct, yeah, or maybe which are not in accordance with your A level syllabus. Okay, so you have to be very, very careful. So I will need to check the video first, and then I'll give you the link. Okay, that's all for the day.